In part 2 of our innate immunity series, we're going to talk about activation of the complement cascade. In our prior video, we discussed how pattern recognition by innate immune cells can lead to the production of inflammatory cytokines. However, complement proteins can also recognize pathogens. And this is shown here, in which a pathogen has compromised some epithelial barrier, and this leads to the activation of macrophages via their pattern recognition receptors. But complement proteins are also present, and these can directly recognize this pathogen and become activated. So what is complement? Complement is a rapid response system to foreign pathogens. It consists of more than 30 inactive proteases. These are found constitutively in the plasma and in interstitial fluid. These proteases are converted to active proteases by recognition of pathogen surfaces. And once this happens, this initiates a proteolytic cascade. Activated complement components have a variety of important functions. One is they can induce bacterial lysis directly. They can also enhance the ability of phagocytes to take up and digest pathogens and pathogen components. And lastly, the complement components are really important in promoting the inflammatory response by inducing local vasodilation. There are 3 pathways of complement activation. The alternative pathway, which is induced when complement components are exposed to bacterial membranes directly. The lectin pathway, which is induced when complement components are exposed to pathogens that are coated with a molecule called mannose-binding lectin. Mannose-binding lectin is typically induced 24 to 48 hours after an infection. And then the classical pathway, which is when complement is activated by the binding of antibodies to pathogen surfaces. And so this typically takes the longest to come about because it requires the production of antibodies before complement is activated. Although I should mention that the classical pathway can be activated immediately if antibodies are already present, either through prior exposure or vaccination. All 3 of these complement pathways share many similarities in how they induce the functions of complement. Each of these pathways leads to the formation of a complement complex called the C3 convertase. The C3 convertase in turn leads to the cleavage of this complement component C3 into 2 fragments, C3a and C3b, each of which have critical functions in mediating the functions of complement. So let's look at the alternative pathway from kind of a 10,000-foot view so you can get an idea of how the complement pathway is activated. I don't want you to get into the fine details of all of the factors that lead to the activation of the complement pathway, but you can use this as a reference as you dig into complement in various circumstances later during your first couple of years in medical school, and hopefully it will give you a conceptual understanding of this pathway. So complement is activated pretty much all the time. So there's a factor of complement called factor D that leads to the cleavage of 2 complement components, C3 and factor B, into 2 subunits, C3b and bb and C3a and ba. C3b and bb bind to the surfaces of membranes and form what is called the C3 convertase. Now, if we are a mammalian cell, mammalian cells have a number of inhibitors that destabilize this C3 convertase and prevent it from activating complement any further. And so normally on a mammalian cell, complement can't really get activated to any great extent. But if it's a bacterial cell, this C3 convertase will become very efficient at now cleaving any C3 in the interstitial fluid into C3a and C3b. C3b will bind to the surface of the bacteria and also lead to the formation of the C5 convertase. So the C3 convertase cleaves C3 and induces the coating of pathogens with C3b, and it leads to the formation of the C5 convertase. The C5 convertase, as you might predict, leads to the cleavage of a complement component called C5 into C5a and C5b. Once C5b is present on the surface of pathogens, it leads to the recruitment of a number of other complement components, C6, 7, 8, and 9, that form a structure called the membrane attack complex. Essentially, the membrane attack complex pokes holes in the surface of bacteria, leading to their lysis. Okay, so the C5 convertase cleaves C5, C5b then recruits the MAC, and the MAC pokes holes in the bacterial membrane. There is another important pathway that I'm just going to mention briefly called the classical pathway. It shares many components with the mannose binding lectin pathway, which I'm not going to discuss in this tutorial. The classical pathway differs from the alternative pathway in that it requires antibodies to bind to the surface, and some of the initial components that initiate the classical pathway differ. However, despite the fact that these components are different, they still lead to the formation of a C3 convertase on the surface of bacterial membranes. And this C3 convertase does the exact same thing that it does in the alternative pathway. It cleaves C3 into C3a and C3b and leads to the formation of the C5 convertase. Once this happens, everything about the classical pathway is exactly the same as the alternative pathway. It leads to the formation of a membrane attack complex and the lysis of bacteria. So that's really all I'm going to say about the classical pathway. The key aspect that you should know is that the alternative pathway is activated by direct exposure of complement components to bacterial membranes, whereas the classical pathway requires antibodies to bind to pathogen surfaces to activate. So what are the functions then that you should know about that complement activates? Those three functions are bacterial lysis, opsonization, and vasodilation. So let's go back to the idea of the C3 convertase cleaving C3 into both C3a, and in this case, we have cleavage of C3a and C5a, but we also see attachment of C3b subunits to the surface of pathogens. Well, it turns out that many phagocytes, including neutrophils and macrophages, have receptors for this C3b subunit. And these receptors are phagocytic receptors. And when they bind to C3b, this enhances their ability to phagocytose this pathogen or eat it and take it up into this structure called a phagolysosome. That is why these C3b subunits are referred to as opsonins. Opsonins are products that enhance phagocytosis by coating the surface of pathogens. The second function of complement is the induction of inflammation. Remember, we talked not just about C3b and C5b, but also the other fragments of C3, C3a, and the fragments of C5, C5a. These fragments can bind directly to endothelial cells and induce their activation and vasodilation, leading to the migration of cells from circulation into the inflamed tissue. So these are critical mediators of the inflammatory response. And lastly, these complement components can mediate bacterial lysis directly. Now, complement is inhibited by a number of inhibitory factors. You do not need to memorize the names of these inhibitory factors. I only want you to know that they exist so that you can understand that complement is targeted towards pathogen membranes and is inhibited on your membranes, mammalian cell membranes. These factors include factor H, factor I, decay accelerating factor, membrane cofactor protein that destabilize the C3 convertase, as well as CD59 that destabilizes the membrane attack complex. So that is a rough outline of how complement works. Again, don't worry about memorizing every component of complement. Instead, focus on those components that are important for opsonization, for the induction of an innate inflammatory immune response, and for bacterial lysis.